This is a production of Cornell University.
Thanks for that nice introduction, Gillian. And as I was preparing for this talk, I realized that it was almost to the day, 41 years ago, that I gave my first research seminar right in this very room. But I think the front of the room was over there in the back at that time. And so I also remember, and this is not a great memory, but like 10 minutes before that, I was down in the men's room there, like hyperventilating and trying to keep from dry heaving because I was just so nervous about giving my first talk. And fortunately, things have gotten a little easier over the years. But anyway, perhaps more pertinent remembrance from that time was also down the hall here. There's a little room that contained a microscope, a beautiful Zeiss microscope. I don't know if it's still there even, but at the time it was the late 70s and it was state of the art. It had differential interference contrast optics, Nomarsky optics. And it just was just a great thing. So, and one of my office mates at the time, Rick Howard, had a project on that microscope. So we were working late one night and he called me down to his lab and said, "Corby, look at this, it's Fusarium." And I looked through there and it was like I was seeing living things for the first time. It was this cell, hyphal tip cell, and I saw a cytoplasmic streaming. I saw vesicles shooting back and forth. And I even saw a nucleus trying to squeeze its way through a septal pore. And it just changed the way I thought about fungi to actually see them. And maybe it's because I'm a visual learner. But that made a huge impression on me. And I've always kept that in the back of my mind, that first time I actually seeing a living cell in such detail. And so about 10 years ago, I guess I started on this project I'll be talking to you about today, and it draws heavily on microscopy, and especially fluorescent microscopy and even super resolution. And it requires this now state-of-the-art sort of imaging to really to make it possible. And I work with mycotoxins, and it occurred to me a few years back that although the biochemistry of mycotoxin synthesis was known, all the enzymes had been worked out, some of the regulatory processes, that no one really knew what was going on in the cell. Where were these secondary metabolic pathways occurring in the cell? So we thought we'd give a look in the system that I worked on, which is Fusarium head blight of wheat and barley. And it's caused by, among other things, this fungus, Fusarium graminearum. And when it infects wheat and barley, it causes these necrotic symptoms. And on wheat especially, it causes this kind of spreading necrosis within the head. It turns out Fusarium graminearum, unlike many Fusarium fungi, really prefers to infect the head instead of the roots, like other Fusarium. And when it does that, it infects the grain and literally causes it to shrivel up and blow away during the harvesting process. But what grain is left behind is often contaminated with these trichothecene toxins. And these are important mycotoxins that we really want to keep out of the food supply. They cause alimentary toxic elukia, which causes neutropenia, which basically does a number on your immune system. And it also causes extreme gastrointestinal inflammation. And that's never good. So what what we've been doing is trying to find ways of preventing this toxin from accumulating in plants. But the toxin also is a virulence factor during infection. So if you make a mutant of the fungus that does not create this toxin, what you get is just a fungus that causes a little necrotic fleck at the point of inoculation. It doesn't spread like this. It causes such dramatic necrotic symptoms. Now, what the Fusarium, oh, I should mention that these trichothecenes are sesquiterpenoid compounds. So they're made up of these 5 carbon units that I'll describe in just a moment. So these sesquiterpene toxins like deoxynivalenol affect protein synthesis, especially during the elongation factor process at the ADS ribosome. And what this does when it produces it in the plant is essentially quell the induced response of the plant. So you get, it allows the fungus to colonize this, this challenged tissue. Now, during evolutionary time, the plants have responded to the presence of this toxin by creating a detoxification system. And these tend to be these UDP-dependent glucosyl transferases. And what they do is basically take the DON and glom on a glucose molecule at this 3 position, and it changes the biochemical character of the molecule, and it allows it to be, oh, thank you, allows it to accumulate in the vacuole and be further metabolized. Now, of course, Fusarium won't stand for that. So what they have done is evolve another sesquiterpene mycotoxin. And what it's very good at is inhibiting this glucosyl transferase reaction. So although culmorin is not toxic itself, it potentiates the disease by making the concentration of DON be greater because it's not then further metabolized. So these 2 mycotoxins really work in concert to cause disease in the plant. So sesquiterpenes and terpenes in general are often primary metabolites. They're used in many processes that are required for all cells to live. And among the products that are essential are things like ergosterol, which is the major sterol produced by fungi. It's important for membrane function. Ubiquinone, which is important for mitochondrial function. Heme and many other primary metabolites are the result of this biosynthetic pathway that goes from acetyl-CoA through the mevalonate pathway, which creates these 5 carbon subunits, which are then combined into farnesyl pyrophosphate. Now, farnesyl pyrophosphate is not only the starting point for all these primary metabolites, but also for these mycotoxins. Now, the mycotoxins that I'm talking about today are not constitutively produced, rather they're conditionally produced to specific molecules and conditions. For example, polyamines are very powerful inducers of these mycotoxins in culture. Low pH is also an inducer. And the most potent inducer is actually just inoculating these fungi into plants. And often like 0.1% of the dry mass of infected tissue can be these mycotoxins. So there's a lot of this being made. Upon induction then, what you get, the first thing you get are these soluble enzymes, cytosolic enzymes, which take farnesyl pyrophosphate and cyclize it. And so this longeborniol synthase makes longeborniol, which is precursor to culmorin, and then trichodiene is the precursor to the trichothecenes. So the other induced components of the pathway tend to be these integral membrane proteins, generally monooxygenases, which can cyclize, but also create these hydroxyl decorations on the molecule. So these are predicted to be endoplasmic reticulum localized. So what does this sort of induction look like in culture? What we did for this experiment was to GFP label one of those cytochrome P450 monooxygenases, Tri1, and then follow it during induction. So we added this 5 millimolar putrescine at 0 hours. And then you can see at about 36 hours in this Western that it appears and then peaks sometime after 48 hours. And you can follow it in a fluorescent micrograph too. So I don't know if you can see these green dots here, but certainly at 48 and 72 hours, you can see this. And this time course matches very nicely to when the mycotoxin actually starts being produced in these cultures. You can also see that there's these very strange structures where the fluorescence is localized, these crescent-shaped structures or circles. And that was puzzling when we first observed them, and I'll get to that a little bit later. But I should also say that the cultures themselves look very different when you induce them. They get thicker, they become heavily vacuolated, and then they branch quite a bit, and they make these large swollen cells or bulbous cells. And I'll mention these toward the end of the seminar. So this is one of the consequences of induction. Now within the cell, the organelle perhaps most altered is the endoplasmic reticulum. And this shows a transmission electron micrograph in a non-induced cell where the ERs primarily exist as rough ER. It's dispersed. It's associated with ribosomes, these black dots. And this is quite different than what we see in these toxin-induced cells, where we get this very elaborate smooth ER, these structures that at times seem to arise from the nuclear envelope. At times they are in the peripheral ER, but they're not associated with ribosomes. And what they are just layers of ER folded back on itself like ribbon candy. So there's just layer and layer of these. And in 3 dimensions, they look like sheets, of course. So you get a better sense of that in these 3-dimensional 3D renderings. This would be the nucleus. So it turns out that those very unusual fluorescent patterns that we were seeing with the Tri1 enzyme, here it is in the pathway, corresponds to this modified ER. So what we did was to label several of the enzymes in the pathway, including a Tri11 now. And they showed cofluorescence to these structures here. And these correspond to the ER based on the fluorescence of this dye-ER tracker, which is specific for ER. In addition, one of the enzymes, but not all of the enzymes of the mevalonate pathway is also found in this structure, as shown here. So because of the pathway enzymes all being localized to this modified ER, we refer to these organelles as toxisomes because they're the presumed site of toxin synthesis. That's where all the enzymes are. The only thing that doesn't really follow that pattern so much was the first enzyme in the step, which I mentioned is cytosolic. It's not anchored to the membrane like these other ones are. And indeed, its fluorescence, at least at this resolution, is consistent with a cytosolic protein, but we'll see in just a moment that it's more than just that. We always try to make sure what we're seeing in cultures corresponds to what we would see during a plant infection. And so, first of all, I should acknowledge Marika Bernish, who's the postdoc who did like 90% of the micrographs in this presentation. Just really wonderful, very careful work. So Marika, for her PhD work in Germany in Willi Schaefer's lab, showed that the infection structures of Fusarium graminearum, these lobate aprosoria, they're multicellular, and those also produce toxin. These are toxigenic cells. And you can see they're kind of swollen and highly branched, just like our cultures induced to produce toxins. So these lobate aprosoria are on the surface of the plant. Then you can see a similar structure in a fluorescent micrograph. And here in red would be the toxisome structure. So each one of these cells in the lobate aprosoria contains toxisomes. Also a ton of nuclei, which is an interesting observation as well. So the toxisomes are forming not only in culture, but also in the plant interaction. And we've also shown that these are not artifacts of the fluorescent protein being there using ER tracker and super resolution microscopy. So let's return to that cytosolic protein Tri5, trichodiene synthase. So what we have here is a super resolution image of a toxisome labeled with one of these monooxygenases. And this is in a single focal plane. You can see that the GFP fluorescence kind of fits over this toxisome as if it were a helmet on a head. And we can do this in 3 dimensions by taking multiple focal planes throughout a cell and then building that into a 3-dimensional model of the cell with toxisomes. And I think you can see how the green fluorescence seems to envelop these toxisomes and really be closely associated with them. Moreover, we can do optical sectioning through those toxisomes and show a cofluorescence of the cytosolic protein with the red membrane-anchored monooxygenases within the toxisome. So what we believe is going on there is that the Tri5 protein really is being captured between the successive leaves of the toxisome. So as the toxisome is forming these sheets folding back on each other, they capture cytosol within them, which leads to this cofluorescence, meaning that they're certainly very closely localized. We want to do that for more than just one cell, because if you look at one cell, you can cherry pick. And so we did a population of cells, I guess 91, and to test whether or not there was an affinity for the toxisome for this cytosolic protein, we measured the maximum fluorescence within 250 nanometers of the toxisome and compared that to the maximum fluorescence of the same enzyme greater than a micron away from a toxisome. And we could express that as a ratio, if it's in the same cell. So a ratio of greater than 1 means that there's a significant enrichment for the cytosolic protein with the toxisome. And indeed, we found that it's significant, and there's many where this is very much brighter than 1. And as a control, we chose another cytosolic protein, which which is also in the cell. And it actually showed the kind of the opposite relationship. So you can really visualize this if you imagine focusing, like you're looking through a microscope and focusing down on the cell. So what we're doing here is just focusing down on these cells, either labeled with the Tri5 protein, where you do see these areas of cofluorescence in every section, versus this protein, where you see this is just cytosolic. There's no apparent interaction. So there is something special about Tri5 being in there. It's not just your run-of-the-mill cytosolic protein. So we're also interested in the relationship between primary metabolism and secondary metabolism. So I said the cells make a lot of these secondary metabolites when they're induced. So when the fungus goes from a non-induced to an induced state, you can imagine there's a lot of carbon flow that would ordinarily just go straight to the primary metabolites. It's drawn off to these other pathways. So how does the fungus adapt to those different pathways occurring in the same cell? So one of the kind of simple experiments we thought to do is, if this was drawing from the same pool of farnesyl pyrophosphate, if we deleted this enzyme, would that then lead to a greater amount of culmorin and its tautomer, culmorin? So what we found actually when we did that experiment is just kind of the opposite, because we thought that it might make more culmorin to have more raw material to make it. But in fact, what we found was just the opposite. So this is a GCMS trace of the volatile terpenes in culture headspace of these cultures, either wild type or the deletion strain, which is in gray, okay? So here in the wild type, you can see the major trichothecene peaks as well as culmorin. But in the deletion, it's basically flatline. So not only does it lose the ability to produce trichothecenes, it loses the ability to produce culmorin, which is a separate pathway. That kind of blew our mind. So we were thinking that the Tri5 protein, embedded as it is in the toxisome, might have some other function, maybe a structural function. So to test that, what we did was to create an allele of Tri5, which was inactive. So it lacks cofactor binding sites. So it's enzymatically dead, but has the same three-dimensional structure as Tri5. So what Chris Flynn did was to add that then back to the deletion strain. And what he found was that indeed this allele restored wild type levels of the culmorin production, as well as some of these other sesquiterpenes as well. So this convinced us that it must have some kind of either regulatory or structural role. And the obvious place to look would be at the toxisome. So in looking at the toxisome, again, with those same three mutants that were in the previous slide, here's the wild type fluorescently tagged strain with the toxisome structures, produces Don culmorin. The deletion strain, this is a HMG-CoA reductase, which is part of the toxisome. And the fluorescence here looks as if it were the undifferentiated ER, like we would see in a non-induced cell. So it's still going to the ER, but it's not forming toxisomes. Then if we put back the inactive mutant allele, the toxisomes reform and we get culmorin. So we believe that the toxisome may also be essential for the culmorin synthesis as well as trichothecenes and maybe other sesquiterpenes as well. our model is that based on the crystal structure of Tri5, it seems to exist as a homodimer. So we're thinking that the Tri5 may actually stabilize the toxisome structure by forming some sort of cross links, either to the reaction complex or to the membranes themselves of the smooth ER. And we now have mutants of sites we think might be important for these sorts of interactions. And this is a project that's been held up by the pandemic. So hopefully we'll eventually get these sorts of results and test this hypothesis. But overall, what this is doing, what this means in the cofluorescence means that all of the enzymes of the pathway are in very close contact with each other. And this may promote biochemical efficiency by having the intermediates all in a small place. But it also may help because, remember, these are toxic. And in theory, they could inhibit the protein synthesis even of Fusarium. So in this way, it sequesters the toxin as well. This is to show you that the actin myosin cytoskeleton is important for the formation of toxisomes. And this is based on the observation that treatment with latrunculin A, which is a F-actin inhibitor, greatly reduces the ability of the fungus to produce toxisomes. You can still see fluorescence here. So this trichothecene protein is still there. It's just dispersed throughout the cell. And what this means is that it produces much less toxin than the untreated cell. There's another Fusarium-specific inhibitor of myosin 1, which acts with actin, and it also has the same sort of effect. So here's this compound phenamicryl. Again, you see some fluorescence here, but not the intensity that you would see in the untreated cells, and a reduced toxin amount. Now, phenamicryl actually is being used in the field in China. They're mixing it with tebuconazole. So this is an azole fungicide, which will kill the fungus, and then phenamicryl will actually greatly reduce the amount of toxin that they see in grain, probably because the toxisomes don't form. So this is kind of an interesting approach where you can have something like phenamicryl, which is not especially toxic, but actually is targeting the mycotoxin synthesis. So I mentioned tebuconazole, which is an azole fungicide. It acts on ergosterol biosynthesis. So we were interested in where is that in the cell compared to the trichothecene pathway? Is it in the same compartment or in a different compartment? Well, it turns out this has been studied in other organisms, and like in yeast, and there it looks like these ergosterol synthesis also is localized to the endoplasmic reticulum, but either to non-differentiated ER or to these lipid droplets. So what has been observed is that the enzymes for its biosynthesis are embedded in the ER membrane. The product is actually within the two leaves of the bilayer of a membrane, which will eventually bleb out and become a free organelle with a phospholipid monolayer, and with its fatty acid tails kind of embedded in the lipid itself, like ergosterol. So we want to observe these sorts of things in Fusarium to see if it's true there. And we have a very efficient way of viewing lipid droplets using this dye Bodipi NL, which is for neutral lipids. We really see a constellation of these lipid droplets, which also fluoresce with ER tracker. Again, showing that these are derived from the ER. And we can also visualize in the same cell the toxisomes. And you can see the complete non-correspondence of lipid droplets with the toxisome, and that would be shown in this lane here, where it's either red or green, but nothing is really co-fluorescing. So these do look like separate compartments. So then we want to see where the enzymes go. And again, this is another project that's kind of come to a screeching halt with the pandemic, but I'll show you some preliminary results here. And this is the ERG11 protein. And using the DIC or Nomarsky, you can actually see lipid droplets as these little blebs here. And this is where the fluorescence for ERG1 seems to be going. So that's consistent with ERG11 being in lipid droplets. Now, ERG1 is a more complicated story. It looks like it's everywhere. This was the first step in ergosterol biosynthesis. It's the squalene epoxidase. So it looks like it's in toxisomes, in undifferentiated ER, and also in lipid droplets. So I don't know if this is an artifact or what, but certainly it's something worth pursuing if these primary and secondary metabolites really are produced in different compartments of the ER. Okay, so kind of the last part of the story is how do the toxins eventually get out of the cell? And we wanted to address this by first looking at some RNA-seq data that we had, comparing non-induced with induced cells. And we had in mind that there would be certain candidates or things that might explain this. One would be these multi-drug resistance transporters like ABC transporters or major facilitator transporters. And when we look at the RNA-seq data, comparing non-induced and induced, there's a lot of candidates to look at. The other possible explanation is that the toxins are being packaged in vesicles and then secreted by fusion of those vesicles with the plasma membrane. So we looked at a number of genes for vesicular transport, and one that really jumped out was this SSO2 protein because of its location and where what it's known to do. So SSO2 is a snare protein. So these are membrane anchored proteins that really are the kind of the target for vesicles, and they allow the fusion of vesicles to the membrane where they reside. And it turns out that SSO1, so there's two major of these T snares in the plasma membrane. One, SSO1, which is essential for tip growth, which we normally think about when we consider the linear growth of fungi. But there's this other T snare, SSO2, which is important for septum formation, branch initiation, so branching, as well as the secondary wall formation, and the expansion of cells behind the tip. So these are found, again, in mature cells in the septa, this SSO2, but they're not found at the tip. As you can see the fluorescence, this germinating macroconidium, they're not out here. And if you remember what I was saying earlier in the seminar, that one of the curious morphological changes that occur upon induction is this kind of subapical swelling, these bulbous cells. So we thought, well, this process must be really accelerated. We know that the gene is highly upregulated. So anyway, Sean Omar in my lab made mutants of SSO2. And it's really one of the most remarkable mutants I've ever seen because it actually grows faster than the wild type. And here you see, this is, I think maybe, I don't know, 3 or 4 days growth, wild type and SSO2 mutant. And obviously there's a pigment difference. But there's also many, many fewer of these bulbous cells per field than in the wild type. So here's the mutant. And so it produces about 70% fewer of these bulbous cells. And interestingly, it also produces about 30 or 40% less DON than the wild type. So probably because it's just not getting out of the cell. And but it's not everything, because there's still quite a bit of DON that is getting out of the cell. So Sean looked at some of these other candidate multi-drug resistance proteins, and to show that they were actually transporting DON, what he did was to express the proteins in yeast, and in a yeast strain which lacks its endogenous transporters. So it's really sensitive to everything, especially DON. So if you just put a vector into that strain and do a dose response curve for DON, or in this case, 15A DON, at 10 parts per million, it's essentially completely inhibited. And if we put in one of these transporters, ABC1, it's quite resistant to those levels of DON, similar to the endogenous PDR5 multi-drug transporter that is found in yeast. So this is the yeast one. Here's actually the Fusarium homologue of that. So the real proof of what they're doing is to delete them in the fungus from which they came. So here you see DON concentration of the wild type. This is in Planta. The SSO2 mutant reducing it a little bit, ABC1 mutant reducing it quite a bit more. And then if you combine those two mutations, reducing it even further. So we think that both vesicular transport and the multi-drug resistance transporters like the ABC1 are important for getting the toxin out of the cell and delivered to the plant, where it can be a virulence factor. So I heard a little bell go off. I guess that means I'm out of time, but I just wanted to, oh, it doesn't? Did everyone else hear that, or was I just hearing? Okay, all right. Well, I don't have to rush then. So just some of the conclusions. So there's this organelle that we've called the toxisome. It's organized smooth ER. And it forms whenever you induce the fungus to produce toxin. It appears to be required for high-level DON production because things that inhibit the formation greatly reduce the accumulation of DON. And this is has potential for chemicals that can really target toxin production. I have a friend who's in the pharmaceutical industry, and he does this computer-assisted drug design. And he looked at these results, and he says, "Oh, we could make a molecule that would be like 100 or 1,000 times more effective than phenamicrol." So the possibility is out there, and if it's much more potent than phenamicrol, then you could add just like very vanishing amounts of this compound to inhibit the production of the DON in the field. So we think that the vesicular transport may do two things: sequester the toxin from the rest of the cell, and then aid in its secretion. But it's not the whole story. That the cells also require these plasma membrane localized transporters, which effectively pump the toxin out of the cell as well. Well, I have a lot of folks to thank. I think I showed pictures of everyone as I went along, although I don't think I mentioned everyone. But especially I want to thank Marika Bernish, who's really helped me develop a cell biology program in my lab. And without her, I don't think I could have done it. And Karen Brose, who's my longtime lab manager who just knows how to do everything. She is just a terrific asset to the lab. And then my many collaborators, especially the imaging unit at the University of Minnesota. And of course, funding sources, primarily USDA-NIFA. So thank you. I'd be happy to answer any questions.
I forgot to mention, thank you, Corby.
Yeah.
Reception on the egg quad following this, packaged food and drink. So questions?
Yeah.
The toxisomes, they were inducible with putrescine, or the enzymes were inducible. What happens? Is it reversible? Can you wash out the putrescine, and do those ER stacks vanish?
So for the people in Zoom, I'll repeat the question, which was, is the process of toxisome formation reversible? If you wash out the inducer, would you lose the that structure? And the answer is, I don't know. It's a good question. There's a lot to be answered. One thing is that if you induce a culture to produce toxin, you see the fluorescence for those trichothecene enzymes in only a small fraction of the cells. It's remarkable. So only certain cells will differentiate like that, not all of them. And when one differentiates, then the chances of the one next to it differentiating increase. So sometimes it begins in one and then moves down the line. But it's also important, in an infection, it would be important, you would think, to regulate. I can't just go in there and gangbusters full time. It would be important to downregulate too. And so what happens next, I don't know. But that's a really good question. Yeah.
Hey, Corby, I just wanted to ask a little more about this potential chemical pathway to suppress the toxisomes. Was that something that targeted specifically for toxisomes and not affecting the other kind of ER organelle type of things, but it would have specific activity there?
Well, it's specific because it only works on the Fusarium myosin 1. So any other fungal species, it doesn't work on. And it's because of the particular amino acid sequence of Fusarium myosin 1. But it targets specific the ATPase, the site of the myosin motor.
Thanks.
Yeah. Mm-hmm. Yeah, Teresa.
This was beautiful, Corby. Thank you. A question. I am fascinated by Tri5. I got an impression that it was a cytosolic protein, but then you have a model that it kind of cross-links the lamellae of ER.
Okay, so the question is, how can the Tri5 be both a cytosolic protein and interact with either membranes or other proteins? Well, there are potential sites where the protein can be modified, so lipid adducts may be possible. And so we've created mutants that change those amino acid residues so that they would not act as those sorts of potential anchors, lipophilic anchors. And we'll see what happens. Yeah, but that's really a major focus of what we're trying to do next. Like I say, we have all the mutants. It's just we need to look at them. Yeah. Yeah. Hi.
Yeah. Thanks, Corby. Sort of a little bit related to Maria's question. Do you know what the inducer is in planta? And is there sort of natural genetic variability in wheat or barley for that inducer? Like that's a susceptibility?
Yeah. So the question is, what potentially is the natural inducer of toxin production in the plant, and is there natural variability for that in the plant? And the answer seems to be that these polyamines like agmatine and putrescine are likely the endogenous inducers. And that's because it's been, and this is all kind of correlative evidence, but because I don't think you can actually eliminate the polyamine synthesis without severely affecting the plant. But what has been done, this has been done by Don Gardner in Australia, his lab. So first of all, he screened all sorts of compounds for their ability to induce. And by far the strongest inducer of toxin synthesis were the polyamines, especially putrescine and agmatine. And then the observation that when you add the fungus, inoculate the fungus to the plant, the biosynthetic pathway enzymes, the genes for those enzymes in the plant are upregulated. And also the amount of those compounds are increased in the infected plant. So all of these, and then they have a biochemical inhibitor of polyamine synthesis, one of the enzymes. And when you add that, then there's less toxin produced in those inhibitor-treated plants. So all of those are kind of correlative evidence that this is really the endogenous inducer of Don. Yeah, Adam.
Because the Tri5 mutation affects both sesquiterpene synthesis and culmorin synthesis. Do you think the toxisomes are evolutionarily ancient and just a structure for making all kinds of things that might be toxic, or do you think it's something that evolved with the genesis of these types of mycotoxins?
So the question is, do we think that the toxisome structure, the smooth ER arrangement, is specifically evolved to accommodate toxin production versus some other general process? Well, when we first observed that, we went through the literature searching to see if anyone else had seen anything like this. And the thing that we found that most looked like it were hepatocytes that were treated with toxic compounds. So that's a cytochrome P450 inducer, like benzanthracene. And in these hepatocytes, you see this elaborate smooth ER. So, and we know that cytochrome P450s are induced in those hepatocytes. So I think what it is the cell just needs a lot of ER because there's a lot of ER proteins to accommodate. And so it's just making ER, and I suppose all cells have that capability if they are induced to produce so many of these types of enzymes. That's my guess anyway. So does anyone in the Zoom audience have a question?
Yeah, my question is with the SSO2 mutant, is the growth inhibition dependent on induction with putrescine?
No. So those, it's actually, it's enhanced growth, and it's present even without
right, I'm sorry,
induction.
yeah.
And it's because it just doesn't branch. It branches very little compared to the wild type. So all its growth is directed outward rather than forming these mycelial mats. And actually, if you grow them in liquid culture, there's no difference in dry mass between the mutant.
Right.
So that suggests that it's just the different morphology of growth that's accounting for that.
Right. Okay.
Okay. Yeah.
Just curious, going back to in planta phenomena, have you looked for the toxisomes in planta? And any other thoughts on what might be going on in plants versus what you're seeing in vitro?
Well, yeah, so as I mentioned before in the question and answer period, not all cells are toxigenic. And that's certainly the case during infection. So on the surface, you have runner hyphae, and those are undifferentiated and do not produce toxin. And They know that based on Marika's work from back in Willie Schaefer's lab. So what they did was to take the promoter, the Tri5 promoter, and link it to GFP. So whenever the induction would be found, it would glow green. So they looked at these, the fungus on the surface of the plant. The runner hyphae were not green, but the infectivized hyphae, the Episoria, were greatly greenish. So that indicates that the toxin was just being produced at the point of infection. But what goes on further into the plant, I don't know. I mean, that, I think it'd be a lot harder to do.
Frances Trail, many of us heard her seminar yesterday, and she talked a lot about the cells, plant cells, with higher levels of silica in it. And I don't know if you've connected those dots at all, if that's any way stimulating to the toxin production.
We haven't really looked at it. So those would be at the trichomes or something like, yeah.
Some other cell types, but particularly trichomes.
Yeah. No, yeah, when those experiments that Marika did were done, it was before Frances's work with silica. So she didn't really address that.
Thank you so much.
Well, thank you for the invitation. It's been fun.
This has been a production of Cornell University, on the web at cornell.edu.